Today, we're diving into immunohistochemistry, or IHC. Now, this is a technique that has totally changed the game in how we understand disease. It's a way for scientists to look past a cell's basic shape and actually see the specific proteins inside, the molecules that tell us a cell's true identity and what it's really up to.
All right, so here's our game plan. First, we'll see why the old-school staining methods sometimes just don't cut it. Then, we'll get into the secret sauce of IHC, the principle of specificity. After that, we'll look at how we can actually see the results, compare the two main ways of doing it, and finally, we'll talk about something that's absolutely critical: validation.
For more than a century, pathologists have relied on stains to get a look at tissue. But here's the thing. Sometimes just looking at the shape of a cell, its morphology, can leave some really crucial questions unanswered, and that's exactly where IHC steps onto the stage.
So take a look at this. What you're seeing here is a breast cancer sample stained with something called hematoxylin and eosin, or H&E. This stain is the absolute cornerstone of pathology. It's fantastic for showing us the basic structure and, yep, confirming that cancer is present. But, and this is a huge but, it tells us that it's cancer, not what kind of cancer.
And believe me, that distinction is everything. Is it ductal carcinoma or lobular? Under a microscope, they can look almost identical, but they behave in totally different ways, and, you guessed it, require completely different treatment plans. Just relying on shape alone would be like flipping a coin on a patient's life.
And this is where the magic happens. On the left, with the standard H&E stain, you've got two breast cancers that look remarkably similar. Now look to the right. Here, we've used IHC with an antibody that targets a protein called E-cadherin, and bam! One tumor stains dark brown, screaming that it has the protein, while the other one is completely negative. That single molecular difference gives us a definitive diagnosis. It's no longer a guess.
The power of IHC doesn't stop there. It also helps predict how a patient might respond to therapy. So again, breast cancer on the left. On the right, we've stained that same tissue for the estrogen receptor. All that brown staining you see in the nuclei of the cells is a direct message to the clinicians. It tells them this tumor is fueled by estrogen, which means the patient is a great candidate for hormone therapy. This isn't just about diagnosis anymore. It's about guiding personalized medicine.
So, the big question is, how does IHC achieve this incredible precision? Well, it all boils down to a fundamental biological principle: specificity.
The whole technique is really built on a beautiful biological concept, the lock and key model. We're using the natural ability of an antibody to find and bind to its one specific target, a protein we call an antigen. You can think of the antibody as a perfectly cut key, designed to fit only one specific molecular lock out of the thousands inside a cell.
And this gets us to the heart of the matter: specificity. For this to work, the antibody has to be like a molecular bloodhound. It has to hunt down its one specific protein target in a sea of thousands of other molecules and bind only to that one. If it latches onto the wrong thing, the result isn't just inaccurate, it's completely meaningless.
Now, the other side of that coin is sensitivity. It's not enough to just find the right target. You have to be able to see it. Sensitivity is the power of the technique to create a visible signal even if there are only tiny amounts of that protein in the cell. For any of this to be reliable, you need both: the specificity to find the needle in the haystack, and the sensitivity to actually see that needle once you've found it.
Okay, so we've used our specific antibody to find our target protein. Fantastic. The only problem is, antibodies are invisible. So how do we actually see where they've gone? Well, this brings us to the systems we use to make these molecular interactions visible to our own eyes.
There are really two main ways to go about this. The first is called immunofluorescence. Here, you tag the antibody with a molecule that literally glows when you hit it with a certain wavelength of light. The second method is chromogenic. In this case, you link your antibody to an enzyme that starts a chemical reaction, which then deposits a stable, colored material right on the target.
Now, to actually see that glow from fluorescence, you need some special equipment. A dedicated fluorescence microscope uses a pretty clever system of filters. One filter selects the exact wavelength of light needed to excite the glowing tag, and a second filter makes sure that the only light that reaches your eye is the light actually being emitted from the sample.
And the result is an image like this one. I mean, it's just striking, right? You get these incredibly bright signals against a pitch-black background. It's perfect for pinpointing a protein's exact location. But there's a downside. The signal fades every time you expose it to light, a process called photobleaching. That makes storing these slides for a long time a real challenge.
The chromogenic method, on the other hand, is built to last. That brown stain is a physical deposit of color that isn't going to fade. You could store these slides for decades. And just as important, you can view them on any standard light microscope you'd find in any pathology lab anywhere in the world. That robustness is exactly why it's the workhorse for clinical diagnostics.
Okay, let's peel back one more layer here. We know we need to attach a visual tag to our antibody, but there are actually two main strategies for doing that, and each has its own pros and cons.
The most straightforward way is the direct method. It's exactly what it sounds like. You take your primary antibody, the one that finds your protein of interest, and you attach the visual tag directly to it. It's a simple, fast, one-step process. But the more common way, especially for diagnostics, is the indirect method. This is a two-step process. First, an unlabeled primary antibody goes in and binds to the target. Then you add a second antibody, called a secondary antibody, which is designed specifically to recognize and bind to the first one. And this secondary antibody is the one carrying the visual tag.
So why the extra step? Well, the main advantage is signal amplification. Because multiple secondary antibodies can all grab onto a single primary antibody, you end up with a much stronger signal. This dramatically boosts the sensitivity of the whole test. Plus, it's more flexible. You can use one type of labeled secondary antibody with lots of different primary antibodies, which is also more cost-effective.
We've seen how incredibly powerful IHC is for revealing a cell's secrets. But with that kind of power comes a huge, non-negotiable responsibility for scientific rigor. You have to be absolutely sure that what you're seeing is real.
It is so important to remember that an antibody is just a tool, and if that tool hasn't been rigorously tested and validated to prove it only binds to its intended target, your results can be dangerously misleading. Poorly validated results don't just lead to errors, they can create what some call scientific myths that can misdirect entire fields of research for years.
So, validation is a whole process. Every single experiment needs to have positive controls, tissues you know for a fact contain the protein, and negative controls where you know it's absent. You have to carefully fine-tune the antibody's concentration to get a strong signal without a ton of background noise. And you have to make sure the way you prepared the tissue didn't damage the very thing you're trying to see. Ultimately, immunohistochemistry gives us this extraordinary window into the molecular world of the cell. It changes diagnosis from just interpreting shapes into reading hard molecular facts. But that window is only clear if we are absolutely meticulous. A technique this powerful demands that we always ask one question. Are we just looking at an artifact, or are we seeing the molecular truth?